All right, so we should be live now. Again, chapter 11 is short, but it's not that it's not important. It's actually really important, especially for eukaryotic organisms. So I'm going to go slow through it, not just because there's not a whole lot of content, just because it is pretty critical. And it's not, the first time I went through it, I ended up going back through chapter 10. I'm like, wait, that doesn't, what, and so now I get it now. I learned some things, which is always good. First time you teach a class, you should learn things, in my opinion, no matter how long you're teaching. So I learned some things. Right? So let's see if we can get you guys can learn some things. I thought let's put together the schedule here. I think let's do. I mean, we could take test 3 on next Wednesday, a week from today. We would be done with the material. I'm okay giving you guys that extra weekend to study if that is okay with you guys. That makes sense, right? It does mean we'll need to start a chapter early, right? So we'll start chapter 12 early. Won't be on, chapter 12 is not on test 3, but we'll go ahead and just get it started early, okay? So that's kind of the schedule. That's roughly what I'm thinking. That I don't see any reason we shouldn't be able to stick to that. So we'll finish. Again, chapter 11 is pretty short. We'll finish it tonight. Review on Monday, this coming Monday, April 5th, for test 3. We'll start chapter 12 a week from today, and then we'll actually take test 3 a week from Monday. And just as a point of reference, last day of withdrawal, that's basically 75% of the way through the semester. That is the week after that. So you guys will actually have a pretty good feel for where you stand in the course at that point, right? I have put out chapters 9 and 10 homework, and there's a chapter 9-10 combo quiz. So it's just one quiz for those 2 chapters. I'll put together, I've got a rough idea on a chapter 11 quiz I'll put out, and I'll probably do just a small chapter 11 homework. I'll get that, and that gives us a chance for you to actually have a chance to look at that homework and get through it before we take that test, okay? So I'll get that put together here in the next day or 2 and get that up on Canvas. So look at that over the weekend, okay? Make sure you're caught up on those homeworks, and that hopefully points you in the right direction for study. Questions here so far? Awesome, okay. So let's see, what else? Something else came to mind it has eluded me, so hopefully it will come back or it's not critical. And we'll go from there. Chapter 11, and we introduced briefly chapter 11 last time. We said chapter 11 focuses on the concept of meiosis. Meiosis is directly related to sexual reproduction, especially in eukaryotic organisms. So sexual reproduction occurs when specialized cells from 2 other parent organisms combine to form a new unique organism. And so we have components from, in most organisms, we say we look from a paternal or from a father, genetic material from a father, and genetic material from a maternal or mother cell combine to form this new unique individual. And then asexual reproduction, which occurs typically in a lot of prokaryotic organisms, a lot of bacteria, that's simply where that organism produces a clone of itself. There are some eukaryotic and some multi-cell organisms that will clone themselves, especially a lot of sea life, starfish, for example, organisms where you can take part of one organism and cut it off or tear it off, and then the 2 sides will now grow into identical clone organisms of one another. Okay, so slight difference, or not slight, significant difference there, where one is we're creating a new genetically unique individual, and the other is just simply a copy. Now, in section 11.2 talks about, well, what are some advantages? Why is that a big thing? If the first organism is good, why not just make a copy of that organism? And we're, to be honest with you, the science is not completely sure and completely in agreement as to why sexual reproduction is what it is. But we'll talk about kind of how that mechanism works. What we do know is that mechanism of sexual reproduction does work almost the same in almost all eukaryotic organisms. Right? And it's the way it's conducted is just the same. It doesn't change much from one organism to the other. They have slightly different ways of doing it, but the mechanisms are almost the same, which tells us that that has likely evolved from a single organism at one point, and that has, through evolution, then branched out into multiple different organisms. The same mechanism is used over and over throughout. It's inherited. Right? That's sexual reproduction. The mechanisms used in sexual reproduction are almost the same in almost all eukaryotic organisms. We mentioned last time this idea of haploid cells. Haploid cells are what we call, gametes are typically haploid or are haploid cells. Right? Gametes are specialized cells used for sexual reproduction. In most organisms, we think of this as the sperm cells from the maternal parent and egg cells from the maternal parent. I should enunciate that, because not maternal parents produce egg cells. Those cells are haploids. Haploids meaning they contain half of the chromosomes of other cells. Diploid cells are cells that contain pairs of chromosomes. So, and these are what we call, another term we'll use there is somatic cells. Especially in animals, animals have a lot of what we call somatic cells. Almost, your text, one of the things I learned is some plant and fungus species, not all of their cells are typically diploid. Some of their cells are not just their reproductive cells are haploids. Some of their cells are haploids. In animals in general, almost all of our cells are diploids, with the exception of the gametes, the cells specifically used for reproduction. All gametes are going to be haploids. You will find some organisms that have other cells within them that are haploids as well, not just their gametes. But again, in animals, typically we find all of our cells are diploids, somatic cells, with the exception of the gametes. Okay. We talked about pairs of chromosomes a little bit. Another term here, so I just wanted to load us up with terminology right up front. Zygote. Zygote refers to the diploid cell, the first cell resulting from two gametes, two haploids combining during sexual reproduction. So in most species, we said it's a sperm cell combining with an egg cell. Each of those has half the number of chromosomes of an ordinary cell. Both of those are haploid cells. So you put two haploids together and you get a brand new diploid cell. And this is the whole concept of sexual reproduction. You're taking genetic material from each parent, combining that to make a unique organism, as opposed to asexual reproduction where we're just generating clones. So again, just some terms there. Gametes, zygotes, diploids, haploids, somatic cells. I think that's most of the terms. Let's see. They might have no, some more. We've defined these already. Okay. Another thing I learned, mitosis, as I was mentioning earlier. There are some organisms where they'll have haploid cells generating more haploid cells. Mitosis generates new cells from a parent cell. It generates two daughter cells that are copies, exact copies. That is mitosis. Mitosis generates copies. Mitosis generates two genetically identical daughter cells from a parent cell. That could be 2 diploid cells, as occurs in animals like us. So one diploid cell copies itself, copies all of its contents, and then spreads. So those are diploids producing other diploids. In plants, fungi, and some algae, you will find some structures in those that are made up of haploid cells, where the haploid cells are producing copies of themselves. I'm not going to claim to be an expert much beyond that. I don't have a lot of expertise in botany. That's plant biology, so I'm not going to speak too much further than that. You can ask questions, and I'll be happy to try to find them out for you. I'm not going to know a lot more than that. All right. So the process of meiosis. Okay, so the process of meiosis. We'll start with a single diploid cell. And the process of meiosis that we have identified and seen throughout science generates essentially 4 daughter cells that are all haploids. So we start with one single parent cell that is diploid. We generate 4 haploid cells from it. We're going to talk about that process of how that occurs. Okay, and so you can see here, normally this thing makes me show to do, maybe it won't, let's see. Yeah. Just a second. I'd like to draw on that slide. Okay. I'm going to go through all this process, and I probably won't use it the rest of the day. Yes, keep. All right, I don't know why it makes me do that. And now it works. All right, so here, saying that this guy right here, is our diploid cell. Same thing here. Saying that these are diploid cells here. Multiple chromosomes. They're in the X shape, right? Because they've just been copied. So when they're copied, we talked about this last time during mitosis. When those chromosomes have been copied, you have 2 chromatids that form together to form an X shape. And that X shape we call a chromosome, or when the X shape gets split through division. One side of the X is a copy of the chromosome. They're both identical copies on each side of that X. When you split those through the mitosis process, then you get 2 sides of the X, and both of those are identical. Those are both also chromosomes now. As opposed to meiosis, where we start with copies of all of these X's. Half of the X's go to one side, half of the X's go to the other side. And the next stage, then the X's get split down. That's what I've got, yes, what I have on this next slide. So, so in human cells, talked about human cells have 23 pairs of chromosomes. 23 pairs of chromosomes for humans. Different species have different numbers of pairs of chromosomes. But for humans, we have 23 pairs of chromosomes, okay? So, since there's 23 pairs in a diploid cell, that means a diploid cell is going to have 46, 23 times 2. So a diploid cell is going to have 46 chromosomes in it. During the S phase, those chromosomes get copied. They form the X. So 2 copies forms an X. And through the mitosis process, they get split. So you start with 46 chromosomes, 1 copy on each side. That's 46 times 2 is 92, so 92 chromatids. After you go through the mitosis process, those get split in half. So I end up with 46 chromosomes, 46 chromatids after mitosis. Meiosis, on the other hand, So after all the chromosomes, it still goes to the same interface initially up front, copies all 46 chromosomes, makes a copy of them. So now I've got 46 times 2, that's 92 chromatids in the first phase of it. Again, most meiosis goes in a couple of rounds. We call them meiosis 1 and meiosis 2. In the first round, we actually cut the cells down. The cells now become haploids. There's only 23 chromosomes in the first round of division. So half of the X's go to one side, half of the X's go to the other, and that gets split up. But there's still X's. That means there's still 2 copies with each X. Each one has 2 chromatids. So in the first round of meiosis, 23 chromosomes with 46 chromatids. In the second round of meiosis, the X's get split up, the chromatids get split up. So there's 46 chromatids, 23 go one way, 23 go the other way. Now I've got 4 cells with 23 chromosomes ready to combine with another human gamete, also with 23 chromosomes, generate a 46 chromosome individual. So again, meiosis occurs in 2 steps. Unimaginatively, we just call them meiosis 1 and meiosis 2. Keep it simple. There are some differences. We've kind of hit on one of the biggest differences we just hit on is the fact that in meiosis 1, we get down to haploid cells, but they're copied chromosomes. Meiosis 2, we split the copies apart. Questions so far? Okay. So the mechanisms and phases for both meiosis 1 and 2 are very similar, and they share a lot of commonalities with mitosis. So what we end up doing is we use many of the same names that we used in mitosis. We use those in meiosis as well. So I don't think I need to write that down. I think I've got that on the slide. So in meiosis, again, meiosis occurs in 2 steps, meiosis 1 and meiosis 2. In both steps, there is an interface. There's a slight difference between the 2. You have interface 1 where you go through G1, you go through your S phase where the chromosomes get copied. So you have to make the X's, make 2 sides of the copy. And then you go through G2 where you make sure all the DNA is good and then it's ready to proceed. And then meiosis 1 is where you actually split the cell apart. And then cytokinesis is where we actually fuse the plasma membrane and cut it in half. Now I've got 2 different cells. And then interphase 2, one of the biggest differences here in phase 2, we don't copy the DNA in interphase 2. We don't need 2 copies going into interphase 2. We're going to split the copies that we have. So again, remember, S phase is where, in mitosis, S phase is where the DNA gets copied, and we form the X-shaped chromosome, 2 copies of that chromosome. There's no need to do that in meiosis 2. We're just going to split the existing X's. Okay. So I pulled this off Wikipedia. I blew it up, and it doesn't look like it blew up very well. It was way too wide to try to fit on one slide. So I cut it at 2, and I cut the meiosis part in 1 part, meiosis 2, and we'll see on the next slide here. But you can kind of see graphically hopefully a little bit of what's going on here. We're going to provide some details here in just a little bit. But the phases, again, trying to point out here the phases of meiosis, both meiosis 1 and meiosis 2 have the same names we saw in mitosis because there's a lot of similarities. So in the prophase, we're packing those chromosomes together, getting them nice and organized so that they're ready to be divided up. And then you've got a metaphase 1 where they're perfectly lined up and they're ready to be split. And you've got anaphase where they're actually being split. And you've got the telophase where they're actually putting them together. Okay? You're ready then, once you've reached the end of telophase, you've organized the DNA back out, and you're ready to cut the cell in half inside of kinesis. Okay, so this is a very similar process we saw in mitosis, hence the reason for using much the same names. And then, again, the goal is to get down to where I've got just one set of chromosomes, not paired. Going into meiosis 2, I still have pairs of chromosomes, right? It's not pairs of chromosomes. I've got duplicated chromosomes. I need to split the duplicates up, where I've just got single chromosomes ready to combine with single chromosomes from the other parent. So, same basic idea. Prophase 2, metaphase 2, anaphase 2, telophase 2 on the side of kinesis. Again, the good news is you don't have to memorize a new set of names. It's the same names and mostly the same processes here. Now, as we talked about, and I mentioned that section 11.2 talks about why do organisms reproduce sexually? Well, in one reason, one hypothesis that kind of exists, and I guess it's kind of a theory at this point, is that you've got your predators and your prey animals, and they're almost in this arms race of trying to outcompete each other and survive. And that lends itself to needing a lot of diversity within the population to try to find what tools either make you the best predator or what tools make you the hardest to catch prey, whichever the case may be. So we're constantly trying to find organisms, trying to constantly find what tools, or at least it's advantageous to the gene pool to try to find the unique gene combinations that provide an advantage. Sexual reproduction provides a couple of mechanisms that allow for diversity within the gene pool. Okay? And I mentioned this already. I've kind of stolen some of my time. The mechanisms for almost all eukaryotic cells, from simple protists all the way up to biggest wells in the ocean, blue wells in the ocean. The process of meiosis is almost the exact same. And so again, that tells us it's evolved from some common organism, and it hasn't changed within all the diversity of organisms that have evolved from that one mechanism. So it's all that to say this is a really powerful tool that organisms found early and have continued to inherit throughout the development of life. Okay, so sexual reproduction provides lots of diversity within the gene pool. How? Well, there's two ways. There's two mechanisms that occur during meiosis that ensure this massive amount of genetic diversity. Okay. We're going to talk about both of these, but feel free to, let's kind of put them up here. There's the basic ideas. Essentially crossover events, and then a random distribution, a random separation of the chromosomes. And we're going to talk about both of these. Try to demystify both of these. I think your text does a pretty good job. I'm happy to try to throw a different angle at it here, and hopefully between the two of them, come away with some understanding of this. But again, it's really, when you kind of think about, man, this hasn't changed in any eukaryotic organism. No organism has figured out a better way to do this. Even with all the organisms we have, science continues to see this mechanism over and over again because of what it does. That tells us it's really important. So the combination of these two events ensures almost every single gamete contains a unique genetic code within its chromosomes that it carries to fertilization. right? So think about if you know, if you have a sibling, or if you know people that have lots of siblings, and you think about there's similarities, but there's always differences, right? My wife is redheaded, blue-eyed, left-handed, which is like genetic unicorn. she unapologetically will tell you she's a unicorn, right? And genetically she's a unicorn, left-handed, red-haired, blue-eyed. Her brother, and she's about average height, right? Her brother is like 6'4". His hair is lighter color than mine, dark eyes, right? Very different. You get them side by side and you can see some similarities in their facial structure, but very different individuals. You put my sister and I, we have similar looks. She's about 5 foot nothing, right? She's a little more than 5 foot. I'm average height. Yeah, I like to say I'm 5'11". I don't know if that's, maybe I'm pushing it there, but you know, she's short. I'm average height. But there are a lot of similarities there otherwise, right? So again, think about siblings where they're getting the same parents are contributing genetic material, but even in that scenario, because of these two events occur, every single gamete, both egg cells and sperm cells, are going to have a different blend of the genetic code from each parent. Okay? So let's talk a little bit about how that occurs. So again, every other cell within the parent has the exact same DNA, right? The exact same homologous chromosomes should be the exact same. And that's the whole, we talked last time about, it's really important that that DNA gets copied exactly the same every time, because one mutation out of place can cause cancer if we're not careful, or at least leads to other mutations which can go out of control. So in mitosis, it's really important that each daughter cell has the exact same genetic code. In meiosis, it's actually really important that every gamete has a different combination of the genetic code. So every organism has two pairs, or has a pair of the same chromosomes, right? I said humans have 23 pairs of chromosomes. We have 46 chromosomes, so 23 pairs. What happens is, after those chromosomes have been duplicated, you get the X shape. So you get two chromatids are joined together. During meiosis, now mitosis, those chromosomes, they don't have to hang out together. They can just go hang out wherever until they're ready to get to metaphase, and they've got to line up, and they don't even have to line up in specific order. They just have to line up and be pulled apart. That's mitosis. In meiosis, the homologous chromosomes will actually group together. So if we number the human chromosomes 1 to 23, and all the both chromosome 1s, the one from the father and the one from the mother, they line up together. They sandwich, essentially. They form this synapsis. And they're giving you this nice diagram here of some of the terms. Okay? Those are important. I don't know we're going to talk about it here, but a protein structure, right? This synaptic complex. We could Google how to pronounce that. I'm not going to be good at pronunciations. I would get, every time I teach the course, I'll get better. These proteins will hold the two homologous chromosomes together. So all the number 1 chromosomes, both chromosome number 1 from the mom and the dad get held together. Chromosome 1, 2 from the mom and dad get held together. And that doesn't occur in mitosis. They don't pair up. But in meiosis 1, those chromosomes pair up because they will swap information. We call this a crossover event. So the exact same segment, exact same length segment, so you see here, part of this blue chromosome will swap with part of this red chromosome. If we call this the one from the mother, the blue, or I guess the father, the blue, and the red being the mother, right? So some of this blue chromosome, after the recombination process, some of the blue chromosome will now hang out on this one chromatid of the red chromosome, and vice versa. Maybe the other chromatids on the other side, maybe they swap information, maybe they swap different information. Crossover events occur differently on each pair of chromosomes. And the cell keeps track of that, which ones have crossed over, but it allows them to swap information. The same length, right? If this one's 1,000 nucleotides long, it's going to swap with a segment that's 1,000 nucleotides with another chromatid. They call these the non-sister chromatids, right? So the sister chromatids are the two red ones from the mother, the two blue ones from the father. The two blue ones are sister chromatids. The two red ones are sister chromatids. The non-sister chromatids are the ones from the other chromosome, the other homologous chromosomes. Those are the non-sister chromatids. So non-sister chromatids can actually swap equivalent information back and forth. Okay. So during prophase, so you can just kind of hammer out a few details here. During prophase 1, homologous chromatids are held together in what is called this Synaptic Complex. They're perfectly aligned, so it's easy for them to swap analogous material. Right? The same segment on one chromosome can swap for the same segment on the other one. So maybe the chromatid from the mother contains a red-headed gene, maybe the chromatid from the father contains a dark-headed gene. During the crossover event, maybe those swap. So you generate then this unique set, this unique chromosome now that can be passed on to the next generation. Unique combination of genes. So again, non-sister chromatids can exchange information. So the idea here is you've got 4 total chromatids there. Now you've got 4 chromatids. 1 on the maternal chromosome, 1 on the paternal chromosome. Sorry, 2 on the maternal chromosome, 2 on the paternal chromosome. You've got 4 chromatids. But what can result and what will result after these crossover events is now you might have 4 completely unique chromatids ready to be split out into gametes after the whole process is over. Right? So every one of those chromatids, one chromatid is going to go into one gamete after meiosis 2. And because of this crossover event, each one of those can be unique. Each one of those can be different.
So we make 4 different chromatids and create one unique child?
So, right. So only one egg cell is going to get fertilized by one sperm cell. But from here, what we're saying is this is initially all 4 of these chromatids are contained in one cell, right? So what we're seeing here, right? All these are contained in one diploid cell, okay? Here. So these contain all the diploids. They contain all the X chromosomes. Half of these X chromosomes will go one way, half of the X chromosomes go the other way, and then they all get split up. So you've got, let's say you've got, here you can see you've got what, 4 chromosomes, so that's 8 chromatids here. So now that's going to split out to where you've got 2 chromosomes in each of these, but 4 chromatids. And then once those chromatids split, those now become chromosomes because they're not joined. So does that make sense? Okay. That's a good question. Yeah, so here, again, we're looking at, this is the parent diploid cell, 4 different chromatids, 1 chromosome and its copy from the mother, 1 chromosome and its copy from the father. What we're saying is during this process, those chromosomes the chromatids can mix information, equivalent information with each other. So part from the father, part from the mother, can mix together and form a new chromatid that's unique. That didn't exist in either the father or the mother. That's a whole chromosome. So you can get pieces of each parent just in that one event. So kind of back to this slide. I put this slide in a couple places, right? 2 mechanisms that ensure genetic diversity. Why is that important? Well, again, it's kind of an arm. We think the prevailing theory of evolution is this arms race. The predators need to stay ahead of the prey, or better at catching the prey. The prey needs to stay ahead of the predators. So we're constantly looking for better tools to do that. So something that allows for more genetic diversity is advantageous. At least that's the prevailing theory. Okay? I subscribe to that. So how do we do that? Well, crossover events is one. And then the random segregation of those homologous chromosomes during meiosis 1 is the other one. So we talked about crossover events. Let's talk about this random segregation a little bit here, if I may. So I've taken a couple of picture from your text. Right, so from prometaphase 1 is where there are the chromosomes or the chromatids are allowed to swap information. Crossover is allowed to occur. Right, prophase and prometaphase 1. During anaphase 1, right, so this is, let's say this is chromosome number 1 and chromosome 1 number 1. Okay, so again, 23 pairs. Let's look at the first pair. So one of oh, come on down. Come on now. Calm down. I know. Okay. So it thinks something's on the screen. You good now? Good now?
Just gotta talk sweet to it.
I sometimes do. Sometimes do. Okay. Where the heck were we? Let's just go look at the order. All right. Okay, so we have talked about crossover events. Yeah, so we're here. Okay, crossover events. That's one. How do we generate genetic diversity? Crossover events is one, where each chromatid going to a gamete could actually be a unique combination of genetic code from the mother, genetic code from the father. But that's still grouped together in two pairs, right? So it's still grouped together in two pairs, right? So 4 chromatids. We got to get it split out to where you get 4 different gametes from these 4 chromatids. They've got to be sorted. During metaphase, between prometaphase and anaphase, there's metaphase in here. I don't know why it's not on this diagram, but between these two, there's going to be metaphase where all these chromosomes are nice and lined up in the middle of the cell. The mitotic spindle, or meiotic spindle in this case, pulls half the chromosomes to one side. Meiotic spindle on the other side pulls half the chromosomes to the other side. It is random, during meiosis 1, it is random as to whether this meiotic spindle will grab paternal or maternal chromosomes. And it's random as to whether this spindle will grab maternal or paternal chromosomes. So you get this random assortment. Every one of them is going to grab the same number of chromosomes and the same types of chromosomes. Okay? But they're randomly shuffled as to how they get split up. So each of these cells is going to get 1 chromosome 1, 1 chromosome 1, 1 chromosome 2, 1 chromosome 2, 1 chromosome 3, 1 chromosome 3. And each one of those chromosomes carries 2 chromatids. Be split up later. The best way I can explain this is think about this. Again, it's a random shuffling, if you will. It's a random assortment. So I was like, all right, what's something random that we can probably relate to? Well, most of us probably rolled a die before, right? Rolled a pair of dice or multiple dice. Maybe probably played a board game. Maybe played craps. I don't know. Most of us have probably rolled at least 1 die before, if not 2 dice. A 6-sided dice, a standard die, 6-sided die, standard die has 6 sides, 1 through 6. You roll it, and you will randomly get a number 1 through 6 showing on the top side of that dice. That's how a dice works. Okay. So if I take 2 dice, roll them at the same time, that will randomly generate 2 numbers for me. Okay. The possible combinations, because there's 6 possible combinations for each dice, for each die, Each die has 6 possible combinations, and you're rolling 2 of them. So the number of possibilities becomes 6. The number of possibilities times 2 events. That's 6 squared, which is 36. And you can see here just a simple table on the internet of all possible combinations. Okay? So 2 events being shuffled, 6 possibilities for each one of those. Now, if we do that with human chromosomes, I've got 23 different chromosomes, not just 2 dice, I've got 23 chromosomes. But there's only 2 possibilities, where the dice have got 6 possibilities. The chromosome, there's only 2 possibilities. There's only 2, right? There's the 2 chromosomes, and I got to pick. One cell's going to pick this chromosome, one cell's going to pick that chromosome. So there's only 2 things to pick from. So it's 2 to the power of 23, and you run that on your calculator, and that is 8,388,608 possible combinations. Okay? This is genetic diversity. It is unlikely that a collection of probably 1 million sperm cells will have the same genetic information. That you'll find 2 sperm cells in 1 million, they're going to have the same exact genetic combinations, just based on this one alone. But you add to it the fact that before they get separated, the chromosomes can swap information, to scramble it up, basically. So they get shuffled between each other, and then they get shuffled when they get sorted out.
I assume the math on that kind of matches up with the frequency of twins. Approximately.
I honestly don't know if twins are genetic or it's just a random happening. I don't know. I would have to look into twins. I don't know off the top of my head.
Twins were that the embryo split too.
That's if you have identical twins. If you have identical twins, you have 2, they're clones, right? And so when the zygote forms, for whatever reason, when the zygote divides, the 2 cells it divides into themselves generate new individuals, right? Those are identical twins. Paternal twins, where you've got 2 eggs get fertilized at the same time. That's another thing. And those are unique, right? But yeah, if you've got identical twins, they are identical. They are genetically identical. They came from the same zygotes. They just split and developed 2. Is that a genetic occurrence? Is there something in the genetic code that increases the probability that that occurs? I don't know that off the top of my head. I don't know if science has decided that. I have to look into that.
I thought that it had, just because the basic genetics said that if somebody in one family has a history of twins and another person from another history has a history of twins, and those 2 get together, your chance increases.
I would have to look. I'm not disagreeing with you.
I didn't
yes.
take you down.
No, that's
I was
okay.
just curious to see if the math kind of matched up on scenarios like that.
I will tell you this. I know in, because I grew up showing livestock, I know in sheep production, I didn't show sheep or raise sheep, but yuck. I know in sheep production that twins and triplets tends to be genetic. And they will actually, a lot of times, if you're trying to grow your herd, you'll pick for twins. You'll keep the ewes that produce twins. I don't know if that correlation's been made in humans. I don't know. But that'd be interesting to look at. And I'm going to make a note of that. You have piqued my interest, but I don't know. So I just know, I do know there are instances where it is in the genetic code. Other questions? Cool. All right, so again, this slide, I was like, this one's kind of important. Again, just kind of keep us grounded. These 2 concepts ensure that every single gamete contains a unique combination of genetic material from both parents. Because they're allowed to shuffle information between each other, and then they're randomly shuffled when they're separated out into from 4 chromatids down to 4 single chromosomes. Questions here? I feel like this is probably like the most important part of chapter 11, is make sure we get this concept in. Like I told you in chapter 10, make sure you're familiar with the phases of mitosis, and know what the names of the phases and know what's happening with the chromosomes in those phases. That's what happens with the organelles and the rest of the cell is important, but that's really kind of the most critical driving force is what's happening with the genetic code that tells the cells how to function once they've divided and they're going to grow again. Right? And here we're talking about, well, how does that genetic information get sorted down into unique gametes that are ready for reproduce? And I just kind of threw this back in here because I thought, well, I threw this at you earlier, and I didn't know how the discussion, you guys have asked good questions and made good points and asked for clarification. I thought, well, I threw this at you earlier, and it might have been a lot earlier, just to kind of reemphasize. Hopefully this helps. Let's look back at this one. Mitosis. We have 46 chromosomes. We're going to copy all of them. So I've got now 92 chromatids. Before mitosis and after mitosis, you split. Okay? So you split the copies up. Versus meiosis, where, again, same thing. You're going to start with, you got the 46 chromosomes. They've got to be copied. So I've got 92 chromatids, 92 copies of that. And those chromatids are going to exchange information with each other, and then they're going to get segregated out. So I'm going to end up with 23, after meiosis 1, I'm going to end up with 20, in human cells, 23 chromosomes. But during meiosis 1, I'm going to have 46 chromatids. And then the second phase of meiosis, those 46 chromatids are going to get split. During meiosis 2, the 46 chromosomes will get split, and every gamete will only have 23 chromosomes, single copies.
That falls under the gamete.
What's that?
Under meiosis, that falls under the gamete specific to sexual reproduction in
Yes.
haploid cells, 23 and 23 versus the regular process of other cells in the human body, mitosis.
Yes. Yes. Yeah, and again, in all animal cells, aside from the gametes, in animal cells, all cells are diploid. Right? So all cells in animal cells are diploid, with the exception of the gametes. Now, again, in plants, some plants, fungi species, they will have structures within them where they've gone through a meiosis process to reduce, and they have haploid structures, and those haploid cells will go through normal mitosis with only half chromosomes, which that's plants. If you like plants and you want to go into botany, I'm not trying to talk you out of that. It's not me. I don't. Right. So I can't talk much more beyond that. But I do want to make that clarification. Is there are some structures within some organisms where they will go through mitosis with haploid cells. I don't know if I've confused you or helped you, but okay. Other questions? What else have we got here? So just, we haven't talked much about meiosis 2. Make sure we clarify that up. Again, same set of steps that we saw in mitosis. In prophase, there's technically a prometaphase in between the two. Metaphase 2, where these chromosomes line up, they're ready to be pulled apart. Metaphase. Same thing happens in metaphase 1. Same thing happens in metaphase 2. Same thing happens in metaphase during mitosis. All the chromosomes line up in what we call metaphase plates. In the middle of the cell, ready to be pulled apart.
Same.
And then anaphase is where those spindles, whether it's the mitotic spindle or the meiotic spindle, pulls the chromosomes apart, and then the cells are ready to divide up. Okay. That's telophase and cytokinesis. And that's what I have. Questions for me?
How do you think the first doctor reacted when they had twins?
Ooh.
Doctor, I have one. There's another
Well,
one coming out.
You got to remember, a lot of children were delivered before they were doctors. So if I'm an individual trying to grow the size of my clan, I'm probably going to be very happy about twins, honestly.
This is jar.
You're also probably thinking you've got a twofold if your wife, when your wife is born in a different era. You know what I mean?
Yeah.
Come out
Yes.
completely different from the rest of the siblings.
Yes.
What if you want to?
All right, guys.